The overall goal of this in vitro human preclinical model of osteoclastogenesis, derived from peripheral blood monocytes, or PBMCs, cultured with breast cancer cell lines, is to mimic cancer cell osteoclast interactions.
This method can help to answer key questions in the field of bone metastasis about the effects of cancer cell and bone cell interaction within the bone microenvironment.
The main advantage of this technique is that it is a fully human preclinical model.
Although this method can provide insight into bone metastasis, it can also be applied to the study of other bone-related pathological mechanisms such as osteoporosis.
Individuals new to this method may struggle because of the variability among healthy donors and in selecting the appropriate PBMC cell density.
Visual demonstration of this method is critical, as the initial PBMC selection and seeding steps require some manual experience before proficiency can be achieved.
Begin by diluting at least 20 milliliters of healthy donor whole blood in EDTA at a 1-to-1 ratio in PBS. After mixing thoroughly, split the blood solution into 30 milliliter aliquots in 50 milliliter conical tubes. Carefully underlay 15 milliliters of lymphocyte separation medium into the bottom of each tube. Separate the cells by density gradient centrifugation, and pool the white mononuclear cell-containing buffy coats into a new 50 milliliter tube. Wash the harvested cells 2 times in 20 milliliters of PBS per wash.